Hi, so I'd like to welcome you to our seminar for non-scientists and a few extras, by the looks of it. Jan is going to be doing our talk today. Jan studied chemistry at the University of Hamburg, followed by a PhD at the Max Planck Institute for Biochemistry, with his thesis work on the structure of the proteasome. He worked briefly as a postdoc in the Max Planck Institute before moving here to the LMB in 1996 to take up an EMBO long-term fellowship with Linda Amos. Jan became group leader in 1998, with his group largely working on the structural and molecular biology of the cytoskeleton in bacteria, and he's made many important contributions to this field. Jan became the joint head of the Structural Studies Division in 2010, and the director of the MRC LMB in 2018. And today, he's going to talk about a bacterial world. Thank you, Jan.
Thank you, Anne. And thank you all for coming. It's a real pleasure for me to see so many of you here. This is really meant for non-experts, so I spot a few people who know as much about this as I do or more, so this is not for you. So my group works on bacterial cell division. It's the basic question of how you get two cells out of one. And I wanted to sort of really zoom out a little bit and try to answer the question why we're working on the basic biology of bacteria and why we think that's important. So let's start right at the beginning of everything. This is a famous picture taken on the 24th of December in 1968, when I was still very young, by an astronaut called William Anderson, and is actually taken by the Apollo 8 mission as it was circling around the moon. And as you can see, this is the first color picture taken in space, and it shows the Earth to be blue. Now, the Earth, we don't know exactly why the Earth is blue actually, but we think it's because there is a lot of water on it and there's a lot of oxygen, and I'll come back to that in a minute. The water is the basis of all of life, and it has really been there since about 4.2 billion years ago or so. The Earth itself is actually 4.5 billion years old. So what this picture is not showing, it's a beautiful picture, but what it's not showing is that the entire planet is inhabited by bacteria who actually started appearing surprisingly early in the history of this planet, about 4 billion years ago. That's a long time ago. And they most likely appeared in the oceans, and that is what likely makes our planet pretty unique in the universe. We humans only arrived about 300,000 years ago. That's 10,000 times later. That is quite a staggering number. So this black slide symbolizes that actually, until very recently, we had absolutely no idea that these organisms even existed. During the Stone Age, the Iron Age, early Chinese history, the Greeks, the Romans, none of these people had any idea that these organisms even existed. And there's a very simple reason for that, which is that they are really, really small. I need some microscope to look at that. And that's exactly what happened at the end of the 17th century. There's a letter written to the Royal Society in London on the 17th September 1683. That's fairly recent, 350 years ago. And this chap called Anthony van Leeuwenhoek, who worked in Delft in the Netherlands, he was perfecting tiny lenses for microscope, and he looked at loads of different things, as you would imagine. When you have a microscope, you look at plants, animal tissue, tears, sperm, as you would expect of a very good scientist, and also pond water. So what he saw in the pond water is shown here. This is one of the most famous pictures in the history of science, because it's truly monumental what you see there. That in the pond water, there are these tiny little living organisms that we have no idea existed. So how tiny are these things actually? I just brought these cute little models of bacteria. This is E. coli. You will have heard this name. We use this all the time in the lab. If you imagine 1 meter, sorry for the non-UK system, if you imagine 1 meter and you divide this by 1,000, then you end up with 1 millimeter. If you divide the 1 millimeter another 1,000 times, you have 1 micrometer. That's the length of that. or the thickness actually. So it's really, really tiny, and you can't see it with your eyes, but you can see it with a microscope. These ones were swimming, so you can see that Löwenhoek actually made this little drawing that they were swimming, and they had different shapes. For example, there's this wiggly one here, Campylobacter. It causes diarrhea, and it was already present in this picture. And then, in fact, our microscopes haven't really changed much. The images of bacteria still look like that today. It is really difficult to overstate the importance of this discovery, because we were always surrounded by these things, we just didn't know that they were there. And this was in the 17th century, and it would take another 2 leaps to really develop what they started, which is the field of microbiology. It's the biology of microorganisms. These 2 leaps were made by Louis Pasteur and Robert Koch. Koch's germ theory was as late as the end of the 19th century, so that's actually very recent. And what they showed is that microbes are alive. They do not appear spontaneously. That seems obvious to you now, but it was not obvious then. They breathe, and perhaps more importantly, they are the cause of some very common diseases. This is called the germ theory. It's a theory in the sense of science. It's not a theory in the sense of general language. It's a hypothesis, or it's a description of reality with a concept, and that scientists call a theory. These discoveries that they made have been brilliantly written up in this book that some of you may have read by Paul de Cruyff, which describes sort of the time from Löwenhoek to modern times, and how all these discoveries around microbes were made. This book actually got me hooked on microbiology, so I can highly recommend it. So since I said that bacteria do not appear spontaneously, no life does, of course, they must have come from somewhere. As I said, from looking at the evolution, we now know that bacteria have been around for about 4 billion years. And we also know from the history of the Earth that the Earth didn't really look like what it looked like today 4 billion years ago. It may have looked a little bit like this. There were no plants, none at all. It was still quite a bit hotter. Actually, there were lots of volcanoes because the core of the Earth hadn't really solidified fully. It looks a little bit like parts of Iceland look today. And it was really under these conditions that we believe that life started and developed quite early, about 1 billion years into it, or 500 million years into it, already into these little organisms that I showed. We don't know exactly what happened, but people are working on that. So this is sort of a very simple version of the tree of life. It starts at the bottom there with the question mark where we don't know. And people like John Sutherland here at the LMB are trying to find out what the chemical origin of this is. This is not organisms, but this is where do the molecules that form life come from. There's some success, as you probably know, and there's some good ideas now of how the molecules started, but there's not really good idea how all of that happened. So out of that question mark, for some unknown reason, 2 different branches of life emerged. One is called the bacteria, the other one are called archaea. Both of them are single-celled organisms, so they are like these, but they are fundamentally different, as we know from sequencing them. So 2 billion years ago, 2 billion years after that, something pretty amazing happened. An archaeal cell on the right and the bacterial cell on the left. They came together and formed what we now call eukaryotic cells. These are the cells that make our own bodies, that make this, make plants and animals and all the rest of it that we are interfacing with normally. And this event, although it's mysterious, is a little bit clearer now because Baum and his cousin actually developed a theory how exactly that happened, and fairly sure that that's actually what happened. So what you should take away from this is that actually in our own cells and our own bodies, we carry the whole evolutionary history of the Earth, because our cells are a marriage of bacteria and archaea. This also highlights something important and amazing. I still remember when I was younger and I was in school that in biology we learned about leaves, about plants, about animals, about yeasts, about flowers, humans, bacteria, and all the rest of it. And while this is all amazing, it's also confusing and bewildering because there's so much detail and sort of a lot of it doesn't really make sense. So I got almost disappointed when I think it was about 12, when I heard about DNA, the molecule of life. You can see the structure of DNA in our own atrium. This is Watson and Crick's model, atomic model of the structure of DNA that was the first structure determined by what then became the LNP. It was still part of the university at the time. This is the basis of all life. It is the code, as you probably know, that determines what a life form will be and what it will do most of the time. This is summarized by Crick's famous, and slightly wrong, a sketch of what's called the central dogma of biology, which sort of emphasizes the fact how central all of this is. And what it says is that you transcribe from DNA to RNA, and you translate from RNA to protein. And it's the proteins where all the diversity is and where all the chemistry happens. Now, we have about 30,000 different proteins. That's extremely complex. Bacteria have about 3,000 to 5,000 different ones. And still today, because we all come from the same origin as I just showed, still today you can see similarities between all of this. So proteins in our own bodies have often similarities to bacterial proteins because all life happens in this way. We all have DNA in us, which then gets transcribed into RNA, which then gets translated into protein. So in principle, we are all the same. We are all one, we are part of one life because we all come from exactly the same question mark, and evolution has just evolved all of this into all sorts of different forms. Well, if bacteria emerged when the Earth was very hot, why are they still around today? They would have come up in a very different environment. First, they will have adapted during this process of evolution I just mentioned, and there are still hot places on Earth, and here you see one of them. For example, in 1968 again, Thomas Brock went to Yellowstone National Park in the US, and this is a hot spring in that park. And what he did is he just took a little bit of liquid from those springs and looked what's in there, a little bit what Löwe did with his microscope. And what he found is an organism that is now very famous. It's the so-called first thermophile, it's called Thermus aquaticus. It's a bacterium that grows in there. And it grows at about 70 degrees. And we now know that there are many organisms that grow at very extreme conditions. There's organisms that go up to 120 degrees. So this is above boiling temperature at high pressure. And there's organisms that grow in acid at high pressures in the oceans, down there. In the cold, some melt to the ice. There's all sorts of stuff out there that's quite unusual, but it's not unusual because this is just what our planet is like. And the most extreme, perhaps, are the ones that grow without oxygen, you would think. Now, this is because early Earth did not have any oxygen. Oxygen didn't exist then as a gas on the planet. So this benign-looking chemical formula caused the biggest environmental disaster this planet has ever seen. As you probably know, this is the formula for photosynthesis. What happens here is that cyanobacteria discover that you can take carbon dioxide, this is a gas from the air, a little bit of water, and make that into sugar. This is the formula of glucose there. So that needs a bit of energy, and that comes from light, and that's why it's called photosynthesis. And this is all great, because basically organisms learned how to make food out of light and carbon dioxide out of the air. And as you know, plants include that now. There's one problem with this, which is the thing at the end. This reaction is incomplete in the sense that it has a byproduct, oxygen gas. Oxygen is actually highly toxic. It makes everything go up in flames, and it also oxidizes iron. And this is why this planet is actually red. So all the iron that's on our planet actually got oxidized by the oxygen gas that the early bacteria produced. So the entire planet essentially changed its entire atmosphere just because of this chemical reaction which these bacteria invented. So some organisms pretend that this never happened, and they still hide in sediments down in the oceans and other places where there's little or no oxygen. So there are a lot of bacteria still around today and archaea that cannot live with oxygen. Yeah, that's the wrong page. So they also hide here. Okay. So because bacteria and archaea can digest a much wider array of foods than we will ever be able to, our bodies actually require them. So the animal here eats grass, as you know, it's a herbivore. And its body cannot actually digest grass. It's not possible. Grass is mostly made out of cellulose. It's a completely water-insoluble material. And our bodies and the cows, their bodies cannot actually degrade it into the sugars this is actually made from. So what these animals have is special guts that harbor Fibrobacter and Ruminococcus, bacteria mostly that have special enzymes that degrade grass into sugar units, which then the cow absorbs and lives from. And that's what makes them special, and that's why dairy is such an amazing thing. So the number of diversity of microbes in the gut is staggering, as you probably know, and probably larger than the number of cells in our own bodies. So our own guts have probably more cells of bacteria than we have cells in our own body. These bacteria do not like oxygen, and that's why the cow's gut and our own gut is an ideal place for them to live and to be carried around. In fact, one can consider this the other way around, that the purpose of our own existence is actually to carry around these microbes and to feed them and to make them happy. So the interplay between this microbiome, that's what it's called, the assembly of all these organisms in our cells, and our own bodies is a field of active research right now. There can be so many microbes in a microbiome because these cells are tiny, as I told you. They're about 1 micrometer, some smaller, some a little bit bigger. But these cells are not simple in the general meaning of that word. Here's an up-to-date picture of the metabolism of E. coli, this organism here. And you can see this is not simple at all. This is about 3,600 different proteins depicted as a little black dot inside in the chemical reaction that this protein will actually engage with. So even these tiny cells are already very complex. They do very many different things. Each of these dots is a protein, as I said, and there are proteins that do many different things. There's proteins that help chemicals go in and out, so you can see at the edge. There are these proteins that do that. They make molecules go through the membrane. There are proteins that help with the central dogma, so there are proteins that transcribe the DNA, that translate the RNA, that degrade proteins. There are proteins that digest food, so these will be outside the cell, but often also inside the cell. There are proteins that make everything run smoothly, and that's needed because you need to organize all of this, you need to react to the environment. And in fact, this is so complex that no single human being is able to understand all of this. And you will realize that that's actually a major impediment to the progress of biology as a science, because we cannot contemplate all of this at once. We cannot understand this at the system level, and the only hope we have for that is actually AI, it turns out. There's one more cool thing in this. There's also proteins that defend against intruders in bacteria. And this is shown here where this is needed, because bacteria get sick too. So here you see viruses on the outside, these sort of stalk-like structures. They're called phages for bacteria, but they're just viruses attacking this poor bacterium here down there. What they do is they actually inject their own DNA into the bacterial cell and repurpose it, so that the cell, instead of doing its thing, it then makes more of these viruses. The cell at the end dies, it explodes, and all the viruses that have already been made on the inside there, these black dots, they actually come out and have replicated without being a living organism by themselves. So they're not alive, they don't have metabolism, they just use these organisms in order to multiply themselves, as viruses do. This causes an endless cycle of warfare, because if one of the two sides wins, all of this stops, because if the host or the intruder get the upper hand, then you only have one of them and you cannot actually continue the cycle. So what happens is that there is a finely tuned balance between the attacker and the victim, so that this is always in balance. So for this, the bacteria developed systems to stop virus attacks. And the viruses in return evolve faster and faster in order to keep up with this so they can still invade. This is a never-ending race. One important outcome of this is actually CRISPR. You may have heard about this in the news, and maybe here in the UNB as well. It is actually a bacterial defense system that bacteria have to stop this happening. But in recent years, it has been repurposed by people like Jennifer Doudna, who jointly won the Nobel Prize for that in 2020, who also founded a company that then uses this bacterial defense system in human cells in order to repair sickle cell anemia, which Max Pierce worked on very early on with the hemoglobin structure. So it's a good example how the machinery that bacteria came up with, such as CRISPR, can be repurposed for other biotechnology applications. So, but of course, microbes are not just fun, interesting, or useful. They're also dangerous sometimes. As was postulated by the germ theory, which was posed mainly by Louis Pasteur in 1860, some of the world's most dangerous diseases are caused by bacteria. Not all are, many are caused by viruses, but many bacterial infections persist today. There's typhoid, there's tuberculosis, there's sepsis, salmonella, many sexually transmitted diseases, and so on and so on. The list is very long. This list from a WHO is actually just a list of the most dangerous ones because they develop what's called antibiotic resistance. And it's really good that here in this building, Lalita Ramakrishnan in the MIU works on TB, which is listed there, and Tanmay Bharat in structural studies works on biofilms in Pseudomonas, which is also listed there as a high priority organism. And Felix Wando works on the host side of the invasion of bacteria inside our own cells, such as caused by Shigella and Listeria. So antibiotics such as Fleming's penicillin promised to eradicate all of this. So in the 40s, we really thought we were done with this. But it turns out that over time, when organisms replicate, they make tiny mistakes, mutations, and these mistakes can cause these antibiotics stop working. And this is a very serious problem. And it's sort of probably the case where evolution is most visible to most people, that it is actually possible for any of these organisms within hours to days to develop resistance that then makes it impossible to treat these diseases with a particular antibiotic. There's often another one, and another one, and another one, but after that there's often nothing. And that becomes very dangerous, and it's very difficult to imagine what will happen if one of these things really starts to run away. So it is clear from this that that one reason bacteria are still around is fast growth. This is clear from this resistance argument that these organisms can only really develop this resistance because they are they're growing fast and they can acquire mutations much faster than we can ourselves. So this is illustrated here in this movie, which shows an E. coli bacterium in a time-lapse movie, and what it shows really is that it doubles about every 20 minutes. So you get 2 cells out of 1 within 20 minutes. That sounds not very fast, but the numbers are actually staggering. So if you take us, we double about every 25 years or so, maybe a bit longer now, and plants double about in tens of hours, and yeast in about hours. So that's much slower. If you take this bacterium, 1 cell, and you grow it for 1 day, and you assume there's no limitation of nutrients, which there always is, but just assume there isn't, then that leads to 5 times 10 to the power 21 cells. That's a very big number. 1 E. coli bacterium is about 1 picogram. That means it's 5,000 tons of cells, right? In 1 day, out of 1 cell. So that just illustrates that one of the real advantages that has always existed is the fast growth of these cells. They can just outcompete pretty much everything that there is in nature. Our immune system, bacterial antibiotic resistance, all of these things in the end are caused by this very, very fast growth. So all of what I said is the reason why we investigate the process of bacterial growth, which is cell division in my group. And the question is, how do you get 2 cells out of 1, and how does this then cause this big problem? So I'm switching over a little bit into things that are a little bit more scientific, but I try to make this as clear as possible. So here on the right, you see this process again is with high resolution. So these cells are these are E. coli cells, and they're about 3 microns long, and they go through this process of division. And there's one particular protein labeled here in a different color, in this magenta color, which is FDZ, which you can see forms this ring structure, which seems to be at the heart of this process where you get these 2 cells constricting, this 1 cell constricting and then dividing into 2 cells. And on the left here, there's a bit of a picture that shows a little more detail. So there is this invagination, as we call it, and this ring in blue. And what the function of that ring is actually to position other proteins in a complex called the divisome at the site where you get these indentations. That is sort of at the heart of this process. As is often the case in biology, when you actually look in more detail, this gets quite complicated. So this divisome thing consists of about a dozen of proteins. You can see FDZ, that's the ring-forming protein there. In the inside of the cell, some of these proteins on the outside, some are in the membrane. So this is actually quite complicated, and this is sort of why biology takes what we do here, because it is complicated and difficult to understand. So what this protein, the divisome, actually, sorry, no, I just explain this here a bit more. So what this divisome actually does is it binds to the ring structure there, and then on the outside, it's actually an enzyme. So it just takes chemicals to make these sugar chains on the top there that are not food. They're just there as a structure, and it's the synthesis of these sugar chains that actually is the process that makes the cell change its shape. So somehow, these sugars are put together, and then also the sugar chains are somehow cross-linked. And what we're doing is sort of twofold. We are looking both in cells directly, how that's organized. So here you see an electron microscopy image of a section of a cell. So this is not transversal. So we're just cutting the cell like this and look at a very thin slice, what that looks like. And then you can see actually the FDZ ring there as the structure on the inside. And in the long term, what we want to understand is, why is that ring actually not? Why is that ring where it is? What position is that? How is that attached to the membrane? How is that? Where are the divisomes? How are the divisomes attached? So all of these are questions that we haven't answered yet, that need to be answered. On the divisome itself, we have made more progress. So Lisa Kassamer and Fusinte van den Ende, my group mainly, has solved the structure a couple of years ago, which is sort of the core bit of the divisome. And you can see here, I think you can see, this is the membrane. The sugar chains are polymerized somewhere here, we don't know exactly where, and the cross-linking is happening somewhere here, and this is where penicillin binds. So while this is beautiful to look at, there's many more questions. Where are the sugar chains? Where do they go? How are they cross-linked? How do you actually get to a situation where then these, what we call the cell wall that this thing makes, deforms the cell? And these are all questions that we want to answer over the next few years. That's all I wanted to say. It's exactly half an hour. This is great. So that really leaves me with the most important slide, which is the acknowledgements. This is super important to me because experimental science is a team sport. It is complicated. It is often very hard work, and it needs lots of people and a lot of time, bits of expertise. And I've always been very lucky to have amazing people in my group, and the current lot is absolutely no exception to that. Thank you. I think some of them are listening right now. I hope. So then I've listed the facilities that my group uses, so I haven't listed all of them. Without the facilities, we wouldn't be able to do much of this. Science is complicated, it needs lots of complicated instruments, but it also needs a lot of expertise for particular methods, and then there's facilities that just take us, make things go a little bit faster. And so we're really thankful for all of that to be available here in the LMB. And again, thank you for providing all of that. And then, last but not least, the LMB is an amazing place, not just because of the science, but also because I think it's exemplary work environment, lots of support outside science that is as important as the science support itself. And I think I've listed everyone there. And I would really like to thank all of you for being such great colleagues and for enabling all of that. Thank you.